Awesome. Well, welcome everyone. This is the last show of the day, if you will. Pritwish and I opened this morning at 8 a.m. That was a little ugly. Um, Pritwish actually opened. He opened at 8 a.m. as the host. And tonight, this afternoon, Pritwish is going to close, which, as we were talking about, is probably a first at AMP for someone to do both the start and the end. But welcome to Biodesix's first corporate showcase here at AMP. I've been coming to AMP for almost 20 years, I mean more than 20 years, and it's the first time actually Biodesix, I've been at Biodesix for 13 years, has actually hosted a meeting here. It's always one of the highlights for me, the corporate showcase, and this time I would say it's even more of a highlight because I get to share the stage with some of our key partners. So Memorial, I'll introduce Tatiana later, Alison Ruse from Thermo Fisher, and then Pritwish Pal from Biorad. So really exciting for us to have this team at our showcase today. So first, by introduction, if you don't know Biodesix, and I know most of you, or some of you at least do, from the faces I see that are actually working with us in different capacities, we have two business units, business lines, if you will, that are focused heavily on science, technology, regulatory, as well as commercial excellence. We have sales individuals that cover the United States. That's our area of focus. And we have two areas in terms of commercialization, one being focused on the lung space. So from a med affairs, if you will, support for oncologists, support for pulmonologists, thoracic, we focus on that segment in the market. On the other side, we're much more open, working with academic research institutions, working with other diagnostic companies, Biorad, Thermo Fisher, for example, and biopharmaceutical companies in providing diagnostic test services and development. Two laboratories, so these two labs report to myself. We are headquartered, though, in Louisville, Colorado, recently Boulder, Colorado, and you can see the number of certs on the lab up to and including our FDA-registered specimen transport kitting facility. We also focus all of our R&D and development services from the Colorado laboratory. The Kansas laboratory is 100% operations, if you will, think manufacturing, scale-up throughput capacity in support of our nodule testing or Notify. So the two tests are shown here. For today, though, I'm not going to focus on what these tests do and how sensitive and specific they are and so on. This is simply to show end-to-end. We also, as many mentioned today, are focused heavily on gaining reimbursement to go along with our commercial footprint across the United States. So all of these tests have Medicare plus multiple private coverage policies which are added to every quarter. All of the tests are also New York State CLEP approved. Myself, I'm the New York State Lab Director for our Colorado laboratory. So a heavy focus on investing in technology, whether it be new test discovery, new test development, improving existing products that we have, so the Notify and IQ Lung product portfolio, adding adjacent new lung tests to supplement those in the market, and then, as I said, in partnerships, collaborating to develop new tests and new indications for use. Partnering strategy in R&D looks like this. So we start very early on. We've had collaborations which are very mature with everyone you see on this side, and we continue to grow our partnership portfolio. And I want to really emphasize these are more than vendor relationships. They actually get very deep into how we work with our partners and our collaborators, whether, as you might have seen this morning when we presented in the Biorad Forum, early access to new reagents, or as we work today, I'll talk about Thermo Fisher's work that we did with them in support of ODX-ET, new assays coming to market and regulatory approval. With Memorial, I'm really excited that Tatiana can represent what we're doing with a translational group at MSK and representing some of the work we've done on the mass spectrometry side, so I'm not going to share myself any of that work. Don't want to steal Tatiana's thunder. And then in the consortia, which is where we validate a lot of what we're working on. So whether it's Friends of Cancer Research, we just shared data from AACR Cancer Research earlier this year, where we looked at 8 different trials and valuing ctDNA. And then more recently with the Foundation, NIH, another consortia, where we're looking at standards in ctDNA, and I have to give them a shout out. This is many years. We started a lot of these efforts in the pandemic in creating new ctDNA quality standards, and this is now just out in JPO, Journal of Precision Oncology, in November here. So big kudos to the group. As you all know, these things take a long time. The platform underlying the strategy I just showed you is multi-omic, multi-technology, if you will. Starting with, in many cases, proteomic profiling, which is agnostic to what markers we're looking at. In the case of monitoring, this is a multi-proteomic application for risk of recurrence that could then identify patients who are at higher risk of disease recurrence and progression occurring. We are developing and readiness by end of year for a molecular MRD tumor-informed assay. So subsequent to identification of higher risk patients, we can go on to profile those tumors, identify bespoke assays, and I talked a little this morning about case studies in prostate cancer. We worked with Memorial to follow patients in that setting with specific assays in monitoring and plasma. So lots of opportunity to plug in technologies along the way to answer unmet clinical needs. So in terms of the pipeline, I know this is one of what we wanted to share today, and again, our panel is going to share deeper details than what is on this slide about where we're going near term. So ODX-ET, like I said, Alison will talk about the work we've been doing with Thermo for many years on validating that panel and advancing its approval with FDA. So amplicon-based NGS, and Alison will describe details. We do have a few presentations for reference, and I actually went in today and updated all of them to AMP25 because they're all being presented in some capacity today. This presentation will live on, so I felt like, well, this is appropriate. The ESR1 targeted test, I did share some information this morning on what we're doing there in partnership with Bio-Rad for a multiplex ddPCR assay, and Prithvish, I'm sure will touch on this as well in his discussion. And then some of the work we're doing on risk of recurrence I shared this morning as well. Very exciting though on the proteomic profiling front. Tatiana will share what we've been doing with VeriStrat in 2 new indications for that test, immunotherapy profiling in MSI high cancers, and then in metastatic castrate-sensitive prostate cancer, where we're looking at the VeriStrat signal being prognostic of outcomes on hormone treatment. And then digital diagnostics is in the pipeline near term. This is not probably the right audience for talking about digital, but in the next forum, we will update on what we're doing there on integration of digital into the pipeline. But I did want to share these results because I know Alison and I shared in CAP 25 what we've done with ODX-ET as an independent laboratory in verifying performance now in the Biodesix lab when ODX-ET was an IUO. This is using ODX-ET against a reference assay, so QIAGEN multimodal, and looking at concordance in a reference VAFs versus ODX-ET. You can see concordance very, very high, 98%, with the VAF ranging from 2.8 to 87%. So very high concordance in tissue tests, and a testament to the work that the Thermo team has put into this test and platform. Really only 2 discordant samples that we continue to work through, but like I said, lots more to come from the Thermo panelists in this talk. So with that, I'd at least like to advertise for Biodesix. We see things as a bright future ahead of us. We've got a proven ability, and if you worked with us in COVID, you know we did this, scale and commercialize our centralized LDTs. We've now moved into distributing in collaboration with Thermo, our first partner for a companion diagnostic, IVD, in the molecular test, and we continue to look at other opportunities to develop there. We've established a framework, and we continue to develop this framework to work with major academic and physician network partnerships focused on rapid test improvements, discovery, development, establishing utility regulatory, and continuing our work in the reimbursement space. Our partnerships really is a cornerstone to our R&D efforts as we seek to leverage global commercialization channels in support of our genomics and proteomics testing platform. So for today, I'll introduce our three speakers in turn, and then I'll get off the stage. And Tatiana, it'll be your turn next. From Memorial, Alison Ruse from Thermo Fisher, and Pritwish Pal from Bio-Rad. Their bios are up here, and I am not going to read them. I can print and distribute flyers, but thank you, Tatiana. You're up first. Appreciate it.
Thank you so much, Gary. And thank you to Biodesix for the invitation to present our work, blood-based proteomics for precision oncology. I will focus specifically in predicting treatment response and patient outcomes. So proteomics can help bridge the gap between genomic alterations and cancer phenotypes by quantifying protein expression, post-translational modifications, and signaling pathway interactions. But that requires a high level of complexity that only in the last decade has been kind of characterized because of the development of novel technologies such as enrichment, protein enrichment methods, also more sensitive and high throughput platforms, and of course bioinformatics that help to really understand better these large data sets. So with all of that, we have been able to now quantify protein biomarkers that are key for precision oncology. But here I want to highlight the plasma proteomic profiling that really enables non-invasive and also dynamic identification of signatures that are linked to patient outcomes and also treatment response. A good example of that is the Biodesix test developed by Biodesix. That is basically was developed using the mass spectrometry data from patients with lung cancer with known clinical outcomes. Basically, after analyzing the whole proteome, they were able to identify 8 regions that have very different protein expression. So they found here in this a representative example, patients that are classified as a Biodesix good in blue basically have very low expression levels in these specific proteins of interest, and the patients in red, Biodesix poor, they have elevated levels of those proteins, and that correlates with poor prognosis and of course, Biodesix good, better prognosis. So basically, they developed an algorithm that was able to identify patients' outcomes based on the protein expression in the blood. And this is an example of a study in lung cancer patients that basically demonstrating that Biodesix can stratify the patient's prognosis in good, better overall survival versus Biodesix poor patients that have shorter overall survival. This is our workflow for proteomic profiling. So we, at MSK, we developed this AI-based tool to basically screen electronic medical records to identify patients that are eligible for our cohorts of interest. And with that, we are able to collect samples, blood samples that are sent to Biodesix laboratory, where the plasma sample is isolated and then processed for MALDI-TOF mass spectrometry technology that basically consists on the separation of the proteins based on their mass charge ratio. So smaller proteins travel faster through the instrument. The spectra data is then analyzed, and the Biodesix algorithm separates and assigns the classification, either Biodesix good and poor, and of course, then we have the report, and this has big implications on the treatment decisions. So with that, we are going to start with the first clinical context that I want to present today. We evaluated Biodesix as a prognostic biomarker in metastatic castration-resistant prostate cancer. So this is the prostate cancer disease journey. Prostate cancer continues to be one of the most common cancer types. Only in the U.S., around 300,000 cases are diagnosed every year, and the patients start with localized disease, but then eventually they develop resistance to androgen deprivation therapy, that is the backbone of the therapy for these patients, and they become metastatic castration resistant. This is the most aggressive prostate cancer subtype, responsible of the death of over 35,000 patients every year. So of course, the drug development efforts have focused on this stage of the disease. Now we have multiple available drugs. However, we don't have biomarkers to really decide which drug is the most appropriate for each patient. This is a snapshot of the first-line mCRPC treatment in the US. So next generation AR inhibitors continue to be the most common drug used for these patients. Specifically, enzalutamide and abiraterone, they are the most frequent drugs used. However, mCRPC, or metastatic castration-resistant prostate cancer, continue to be uncurable. So only median survival of less than 3 years. Only 42% of the patients make it to the second-line treatment, and over 90% of the patients develop bone metastasis. So, and that have a big impact on their patients' quality of life. So, of course, there is a huge need for biomarkers to guide personalized treatment in metastatic prostate cancer. So to address this gap, we evaluated whether Biodesix can stratify mCRPC patients, starting treatment with the hormone therapy that I just mentioned, enzalutamide and abiraterone. So we analyzed a cohort of 50 mCRPC patients. So first, we did analytical validation. We compared matched serum and plasma samples collected before the patients starting treatment with either enzalutamide or abiraterone. And we confirmed that the Biodesix classifications have almost perfect correlation in these sample types, in the paired samples. So this was really important for us because it provides flexibility for clinical implementation. Then analyzing the data, 82% of the patients were classified as Biodesix good, 18% received the poor classification, and then we see in the Kaplan-Meier curve, the patients that were classified as Biodesix poor has shorter overall survival with only 130 days versus the Biodesix good patients with 633 days. And then we performed a multivariate regression analysis adjusting for prognostic biomarkers, all of them baseline, of course. So the biomarkers we used was PSA, LDH, and alkaline phosphatase. And as you can see, Biodesix was superior predicting the prognosis of these patients compared to the clinical biomarkers. So definitely an interesting result here. And now let's switch gears to our second clinical context. We evaluated Biodesix proteomic signature for pembrolizumab response prediction. So as we know, immune therapy has revolutionized the cancer treatment. And here I'm going to focus on specifically in anti-PD-1 inhibitor, pembrolizumab, that acts by blocking the interaction between PD-L1 protein and the PD-1 receptor that releases the T cells from that inhibitory signal and allows them to kill the tumor cells. So pembrolizumab was approved in 2020 based on the results of the Keynote 158 and received the approval for patients with metastatic solid tumors that has microsatellite instability high, DNA mismatch repair alterations, or tumor mutational burden high status. However, when we analyze the data from this phase 2 trial, we see that the overall response rate was only the 31%. 1%. And more surprisingly, the response rates varies tremendously across tumor types. With pancreatic cancer, only 18% response ratio, compared with the endometrial that was near 50%. So with considering this data and the data that we have accumulated through the years of using this drug, we see that MSI high, TMB high, or DMMR alterations can be indicators of potential response to pembrolizumab, but they are not sufficient to select patients. So definitely there is a unmet clinical need for reliable biomarkers to predict resistance or response to pembrolizumab. So to address this gap, we analyzed a cohort of 57 patients, all of them initiating treatment with pembrolizumab alone or in combination with chemotherapy. The table is only showing like the proportion of patients with MSI high alterations. That was 77%, and the rest of the patients have alterations in one of the genes from the mismatch repair pathway. This is our workflow. So basically, we collected samples from across 10 solid tumors. All of them, the samples were collected baseline, so before the patients started treatment with pembrolizumab. And of course, we are following up these patients during their treatment, but for now, we are going to focus only in the baseline signal. This is the cohort breakdown. Colorectal and endometrial were the most, like the largest subcohorts within our study. And our cohort is a hypermutated cohort. You can see here the TMB status. The threshold is usually equal to 10 or over 10 mutations per megabase. Our cohort, most of the patients have higher TMB with a median of 38.2 mutations per megabase. So very like hypermutated. The results, so the baseline plasma samples were analyzed, and we see that 72% of the patients were classified as a Veristat good, 25% were classified as a Veristat poor, and 4% had indeterminate classification. That means that the spectra data didn't meet the criteria for these major categories. But when we analyze the time to subsequent treatment or death, definitely Veristat poor has shorter time to the next treatment, or they die with a median of 269 days. Similar with an overall survival analysis, here Veristat poor or indeterminate patients have a shorter overall survival versus Veristat poor, good. Finally, we wanted to evaluate whether there is any correlation between genomic landscape and Veristat classifications. For that, we use the data from our in-house NGS panels, MSK Impact, that is a tissue-based assay, and MSK Access, that is a liquid biopsy assay. And we found that Veristat good patients have higher prevalence of the KMT2B alteration. This is an interesting result, as these alterations of this epigenetic regulator have been associated with increased sensitivity to checkpoint inhibitors. In contrast, JAK1, T53, and KEAP1 have a slightly higher prevalence in the Veristat poor cohort. So definitely these are intriguing results that require further validation, but it will be really important to evaluate whether Veristat classifications can indicate or give us some insight in the tumor biology at baseline. And just to close the presentation, the key takeaways. So first, I hope I show you that blood-based proteomic assays can detect baseline biological characteristics that can help us to predict treatment outcomes. Also, Veristat is a prognostic biomarker in prostate cancer and is independent of the typical clinical prognostic markers. Then Veristat is also prognostic for overall survival and treatment, time to treatment or death in a pan-cancer cohort of MSI-high tumors, initiating treatment with pembrolizumab. As next steps, we are planning to conduct a prospective clinical trial on prostate cancer to validate the value of Veristrat in this disease. And also we are, of course, expanding the MSI-high cohort to validate our results and perform tumor-specific analysis and validate our data. With this, I would like to thank my colleagues at MSK and, of course, our wonderful collaborators at Biodesix, and thank you for your time.
Okay, so thank you everyone for sticking around for the last talk of the day. And thank you to Gary from Biodesix for the invitation to present today an overview of the Oncomine DX Express test. So this is an IVD for rapid genomic profiling. Like Gary gave a nice introduction, my name is Allison Roos. I'm Director of Medical Affairs within the Clinical Sequencing Division at Thermo Fisher Scientific. And I'll try to keep it light so that it is the last talk of the day. So I just want to start with a little background. We know that precision medicine has come a long way. Over the past 2 decades, we've seen an expansion of targeted therapies, and this really was driven by biomarker discovery and drug development. So as you can see on the left, the percentage of clinical trials incorporating biomarkers have increased from 18% in 2000 up to 61% in 2020. And this really has fueled the approval for targeted therapies when in 2020, nearly 40%, over 200 were approved. So biomarker testing is absolutely critical for this process. It's critical for clinical trials, it's critical for companion diagnostic development, and it's critical for post-approval testing of patients to identify those who might benefit from corresponding targeted therapies. So we know that next-generation sequencing truly is a key enabler of tumor profiling. It enables simultaneous analysis of multiple biomarkers in a single sample. You can put multiple tumor types together from patients in a single run, and at the end you get a clinical report. Oops. I didn't even know this worked, to move forward and backwards. And at the end, you get a clinical report identifying either a positive result or a negative result and whether or not that patient may benefit from a targeted therapy. So what this does for a laboratory, it really streamlines the process in their workflow from diagnosis to treatment selection. While this sounds great, we know despite a decade of progress in precision oncology, patients still are missing out. So this was a real-world study that looked across the biomarker testing pathway and really asked the question, where are we losing patients? And what you can see is that from this study, less than 50% of patients that are eligible for targeted therapies actually receive them. And they're lost due to gaps in the testing process. So whether that be tumor insufficiency, long turnaround time, or other factors that may impact their ability to get a biomarker test result. So this is striking. So patients are missing out on potentially therapies that may improve their outcomes. So what we know is that the reality is, thinking about the previous slide, for a lab, you kind of have to make do with what you receive. And so the reality is that, there is a need in, laboratories to test small biopsy samples. So this was another real-world study that looked at, 30,000 consecutive tissue biopsies that came into a lab. And what they were looking at is, what is the amount of tissue surface area that we're getting from these samples? And what they found was nearly 60% did not meet the criteria that's set forth by many labs out there. They're standard, greater than 25 millimeters squared. And so they wouldn't have met that upfront criteria for testing. However, this lab used an optimized test approach using amplicon-based testing, And 94% of those samples actually produced a successful report. And so this talk isn't intended to go through the benefits and limitations of different testing approaches, but I think one benefit of amplicon testing is the ability to have a high success rate on minimal sample input. So our vision at Thermo Fisher was really to enable rapid next-generation sequencing. We knew the barriers that were presented earlier. Turnaround time really can impact a patient from being eligible either for a clinical trial or targeted therapy. Additionally, we know that, like in lung cancer, for patients with metastatic disease, upfront targeted therapy improves overall survival for patients. There are biomarkers that are contraindicated for immunotherapies. So having these rapid test results is absolutely critical. And so what we envisioned was a process where you have rapid next-generation sequencing, which could produce a report within 1 to 3 days. And then at the end of that process, you have an integrated report for the oncologist to make an informed treatment decision. So how did we do that? So today, what I'm excited to introduce to you all is the Oncomine DX Express test. It's along with the system, the Genexus DX integrated sequencer and the purification system. So in July, we received FDA approval for this test, and it was approved both as a companion diagnostic and it also has associated tumor profiling claims. So just to go through those two different approvals, so the test was approved as a companion diagnostic to identify patients with non-small cell lung cancer with EGFR exon 20 insertions that may benefit from Xigrophy. So that's the companion diagnostic claim. The test also detects tumor profiling biomarkers that are recommended by professional guidelines across multiple different tumor types. And so within that tumor profiling claim, you can see it's broken up into a DNA component and an RNA component. The test tests both. We know RNA is really critical for testing for fusions. So on the DNA side, it tests for deletions, insertions, and substitutions. You can see all the genes listed here. The test also detects copy number variants across numerous different genes, and then RNA and splice variants in 18 genes. So, in terms of the coverage, it detects clinically relevant alterations across non-small cell lung cancer, covering most of the guideline recommended genes for upfront testing, cholangiocarcinoma, thyroid, and you can imagine those are tumor types where you tend to receive minimal amounts of tissue for testing. So how does the test work? So there's two instruments, like I mentioned earlier, the Genexus DX purification system and then the Genexus DX integrated sequencer. And so the benefits of this for a lab is automation. So with as little as 20 minutes of hands-on time, it could generate results within 24 hours. That's from specimen to result. And so this workflow really enables that rapid NGS that was our vision that I presented earlier. So if we go through the steps in the process, the day one starts with your typical processing of FFPE specimens. This test is intended only for FFPE specimens. And so you go through typical processing. First step is you put the sample into the Genexus DX purification system. With this approval, you can use other IVD purification extraction kits, so that is allowed, but this is truly an automated purification system. So after the DNA is purified and quantified, the next step is you take that plate, you put it into the Genexus DX integrated sequencer, and this is where the rest of the work happens, basically. So library preparation, sequencing, analysis, and at the end, you get a clinical report. So all in all, 2 days, 2 touch points from sample to report within 24 hours. So with this automated workflow, what that enables is free up the lab to do other things, right? You're taking out one component of their workflow that enables you to do other things, research, other testing that needs to be done for the clinical workflow. And so this is what enables that rapid turnaround time. So as part of our FDA approval, multiple both analytical and clinical studies were performed to establish the performance of the assay. And really, this was a robust validation. And so I'm not going to go through all of these studies. This was covered in an earlier talk today where one of our R&D experts went through all of the different studies that led to FDA approval. But just to go through a couple and throw out why this is important. So we did multiple studies to establish limited detection across multiple different variant types, just to make sure that the test can detect different types of variants at different allele frequencies. Knowing what comes into the lab could differ based on age and different components, storage, processing. We had to test, tissue input. We did a lot of tumor content studies to make sure we can call different variants at different tumor content percentages. Looked at tumor necrosis to see how that might impact the performance of the test. Slide stability, knowing that slides may be a little bit older. So all of these tests were done as part of the analytical studies. And then importantly, because it's a companion diagnostic, we had to perform clinical studies, both clinical accuracy studies for EGFR exon 20 insertions, which is our companion diagnostic claim, and then clinical effectiveness studies. So as part of being a companion diagnostic, you really have to establish that clinical efficacy to enable that approval. So I always feel bad presenting this slide because I didn't do any of this work. And I actually feel especially bad today because the people who did are in the room. But just to give you an idea of the breadth of the studies that were performed to enable the FDA approval, 31 analytical validation studies were performed. The lab technicians and my colleagues, tested over 12,000 DNA samples, 14,000 RNA samples, over 1,500 sequencing runs. And that always just blows my mind. And, kudos to the team who've been there from its infancy and that led to the development in July. One study that I did want to touch on, just because of the importance and the definition of a companion diagnostic is to identify patients who may respond to a therapy. I did want to briefly touch on the performance, the clinical studies. So in order to establish assurance of safety and efficacy of this test to identify patients with EGFR exon 20 insertions, we did a clinical bridging study. And basically what this was, we compared the ability of ODXET to identify patients, the positive patient cohort. And so as you can see in this study, and the data shown here, both the patients within the clinical trial and the ODXET positive population had very similar response rates. And so what this demonstrates was that the overall response rates for the ODXET EGFR exon 20 insertion positive population aligned with those observed in the clinical trial cohort from the clinical study, which led to the drug approval. So I just want to touch on the report because at the end of the process, you get a clinical report. So this is what the report looks like on the right here. So the way the FDA works for classifying these results is they have 3 different levels based on clinical significance. So level 1 in red here, you can see are the companion diagnostic biomarkers. Right now, that is EGFR exon 20 insertions. And you can see the identification of an insertion here and the associated targeted therapy. Level 2 are cancer mutations with clinical evidence of clinical significance. So within evidence within a tumor type and guidelines within a tumor type. Those are the variants that you would find in the level 2 section of the report. And then level 3 are cancer mutations with potential clinical significance. So maybe have data in another tumor type or have, are involved in clinical trials. So this is powered by our Oncomine knowledge base, an expertly curated proprietary database of biologically and clinically relevant biomarkers. So just to quickly touch on how this can help a lab, we know that there are multiple barriers for a lab to bring in an NGS test. So Because this is a validated IBD solution, that requires the lab only to do a verification, which is much simpler and more streamlined. Automation, we've heard a lot about that today in different workshops. Labs, there's an increased burden on them due to the growing number of targeted therapies, antibody drug conjugates, things like that. So 20 minutes of hands-on time, allows a lab to implement this. Rapid results, went through that. You can get from sample to report within 24 hours. And then it has minimal input requirements of only 10 nanograms DNA and RNA, enabling a testing of more samples that might come through a lab. So in summary, with this approval, it enables implementation of NGS and the ability to consolidate oncology biomarker testing. We hope this will enable NGS to be offered in more labs, and you can deliver rapid results to accelerate precision oncology treatments. So I want to thank you and turn it over to Prithwish.
Thank you, Allison, and thank you, Gary, for the invitation to talk at this session. And I know this is the last talk between you guys going out to your dinner plans and your happy hour plans, so I'll try to keep this quick and go through some of the work on droplet digital PCR. So basically what I'm going to talk about is Allison already mentioned, like the importance of biomarker testing, and in fact, my first slide was about these biomarkers and why they're important, but she explained it much better, so I could totally skip over this slide. But biomarkers are important, we know that, so we need various different tools so that to be able to measure biomarkers and help patients. So we've heard about proteomics, we've heard about NGS, and so what I'm going to talk about right now is about droplet digital PCR. My boss is right here, she was about to correct me. Sorry, it's been a long day. So what I'm going to do is talk a little bit about the background of droplet digital PCR, and then cover very broadly at a very high level the portfolio that Bio-Rad has to serve precision oncology, and talk about some of the data that we have generated with our collaborators on a number of different solutions. For those of you who are unfamiliar, so I'm sure everybody's familiar with PCR. So think of digital PCR as the third generation PCR technology. So you have the endpoint PCR, then you had like back in the 80s, quantitative PCR came along towards the turn of the century where you could actually now start quantitating. But then as you can see from this diagram, like in those bulk PCR reactions, you're actually amplifying everything together, and so your background is pretty high. So what digital PCR did was partition all the DNA or RNA into small, like individual molecules, and then amplify those individual molecules and read them. So that, and I think the diagram here kind of does a really good job of showcasing how it kind of gets into like an enriching of the scarce rare variant and to be able to measure that. So that's digital PCR in general. Droplet digital PCR is a technology that Bio-Rad pioneered back in early 2010s, and it's been about 13 years now, where you actually use an oil droplet to put your reaction and your DNA in. We can talk a little more details about it later if you're interested, or you can come to our booth tomorrow to see some of our platforms. But in very short, what ddPCR enables is high sensitivity, precision, and accuracy. It also enables quantification, actually absolute quantification, so you don't have to do a reference curve as you have to do with RT-PCR or qPCR. Some of the new chemistries and some of other kind of secret sauces that we have enables higher level of multiplexing as well. So if you're thinking of PCR as being like, oh, you can just look at just one variant, you can actually do much more, multiple variants with this, and I'll talk a little bit about that. And it provides a easier workflow. Again, as it's been talked about, that workflow is really important, fast turnaround time, cost, et cetera. So In particularly, we take where droplet digital PCR is really useful. It's for liquid biopsy applications. So I'm going to spend a minute on this particular slide. So you have NGS. We heard about NGS just now. Obviously, it provides high level of sensitivity, but the cost, even though the cost is coming down and workflows are getting simplified, the cost of NGS, as well as the data storage and all that, still adds significant, lack of better word, cost and complexity. But if you look at digital droplet PCR, especially if you're not looking at a wide breadth of coverage, if you're looking at a very targeted set of genes or variants within a particular gene, droplet digital PCR actually gives you higher limit of detection and therefore higher sensitivity, and at a lower cost and slightly easier workflow as well. Compared to qPCR, which has been around for a while, again, droplet digital PCR has a higher sensitivity, and it can also offer higher multiplexing ability. So we think for certain applications, and we'll talk about a few of those in a few minutes, droplet digital PCR is a really good fit and can be a complementary technology to qPCR or NGS as your requirement of the particular lab might be. So, and just to mention that we're talking about liquid biopsy, so liquid biopsy has a number of different applications, all the way from screening to monitoring and surveillance, diagnosis and therapy selection, et cetera. And so ddPCR can be used in any of these different stages of the patient journey. A little bit quickly about the workflow. Again, it's a relatively straightforward workflow. Depending on the particular platform you're using, the assay you're doing, it's about less than 8 hours. So you also get a same-day result here. You initially do a partitioning and amplification of the extracted DNA and RNA, and the original source can be plasma or FFPE. You then do the amplification, do the reading, and the analysis. It comes as convenient scatter plots, and you kind of draw like kind of circles around that or polygons around that, and you can subtract the background and get your variant allele frequency, et cetera. So as I mentioned, you can multiplex higher, and we'll talk a little bit about that. Sensitivity, we can go down to 0.01% variant allele frequency using certain chemistries. There are a number of different instruments that we have. So this is kind of showing the different cartridges and instruments that can be used, and you can use different types. And in fact, we just launched a couple of almost automated instrument, like a walk-away instrument where you add your reaction mix and you can walk away, and the instrument does everything for you. Those are the QX700 and the QX Continuum. And again, if you come to our booth over the next couple of days, we can show you some of those instrumentation as well. So depending on the instrumentation, you can get different levels of automation. But again, we do realize that, this is kind of repeating it, that just having a platform is not enough. You have to have content on it. You have to provide solution. So again, going back to this image, and when I gave this talk this morning, I mentioned that this image will show up multiple times throughout the day and throughout the conference. So I haven't kept account, but it's probably somewhere around 100 right now. But along the cancer patient's journey, as you can see, like the level of ctDNA can vary, and you can do early cancer detection. So this is where like the patient hasn't developed cancer, but you're kind of trying to see if they will in the future. So colorectal cancer is a really good example of this, and there's a bunch of other companies that are working on multi-cancer early detection in this space. And then there's diagnosis and therapy selection, which we've talked about a little bit here, as well as then MRD and surveilling recurrence. So the point we wanted to make here is that we do have solutions across this patient journey. So we have, like, you can do methylation detection. I'll actually have a slide on that. You can do SNVs, copy number, gene expression, et cetera, and those can be used, again, across this entire patient journey. In terms of the products that we offer, the ones in greens are ones that are already available. We are working on an MRD monitoring solution, which I'll have a slide about that in a second. On the early cancer screening, and we are not developing anything ourselves, but we do have partnerships and collaborations where we work on that. So it's public knowledge, like we have a collaboration with Geneoscopy for early cancer detection of colorectal cancer, which is not based on liquid biopsy, but it's still stool-based, but it's used for cancer screening. And in terms of the solutions portfolio, so we have three different ways of thinking about the solutions that we have. One is for a specific tumor, if we select the biomarkers that are being looked at, and we develop a particular panel for that relevant to that tumor, and we kit it, and you could just use the kitted solution for that particular types of tumor. We also have what we call a digital assay portal through which you can find individual assays. I'll actually skip to the slide quickly. So these are individual assays that are all the common somatic mutations in the COSMIC database. We also have copy number variation and gene expression assays as well. So basically what this allows you to do is go to the site, and if you have a favorite particular biomarker you're looking for, you just type it in and it'll show whether that assay is available. And a lot of them have been wet lab validated, and the rest of them have been in silico validated to work. We also work with collaborators, both academic and commercial collaborators, to do custom design assays as well. And then some of them we also offer like that you can order directly through us as well. So for this talk today, I'm going to talk a little bit about some of these kits, the MSI assay kit that we have, as well as ESR1, and a little bit about the MRD, as I mentioned. So how are we doing for time? 10 minutes? Oh, I thought we were finishing at 50 past. Okay. All right, so I can go a little slower. All right, so the ddPCR MSI assay. So as you guys know, MSI is a biomarker, and Allison talked about it a little bit. Actually, both of you guys talked about MSI being important for immunotherapy drugs. So there is a pan-cancer approval for Pembro, as well as in colorectal cancer. There are certain approvals for MSI high patients. So usually the gold standard method so far has been with tissue and doing capillary electrophoresis with PCR, or looking at the MMR status with immunohistochemistry or NGS methods. So we developed an assay which looks at 5 different markers. The one thing that's important here is that this assay can be used both for tissue and plasma. So this is one of the few assays that are out there that can actually be used for plasma samples to look at MSI status. And there's emerging evidence that it's beneficial to actually look at MSI from plasma in addition to tissue. The data that I'm showing here this is when we launched the assay. We did some studies with some collaborators in France, where we had 2 different colorectal cancer cohorts. And as you can see that with those cohorts, like both with plasma and tissue, we're getting pretty good concordance and good overall specificity and sensitivity. We also did a study with a group in China where they were looking at both colorectal cancer with their gold standard assays, as well as endometrial cancer. Again, as you can see, pretty good concordance for both these different cancer types. And again, I'm not diving into the details of these, but happy to kind of stick around afterwards if you're interested. And we have other people in the room here who have worked with these collaborators, so can give you more insights into it. This is my favorite part, which is the ESR1 assay. And if you've spent the time today or for the last 1 year, ESR1 is really there's a lot of buzz around it, and the buzz has been created by the SERENA-6 trial that was from AstraZeneca that was presented at ASCO earlier this year. But just to back up a little bit, so the ESR1 mutation, it causes acquired resistance to hormone therapy in advanced breast cancer patients who are HR positive or HER2 negative. It occurs in about 30-40% of these patients who are on aromatase inhibitor therapy. And there is this new class of drugs called the oral selective estrogen receptor degraders, or the oral SERDs, that can target the ESR1 mutation, and patients respond to it, and it improves, provides survival benefits. So the first drug was approved, elcestrant. This is from Maronini. This was approved back in 2023 by the FDA, and then EMA the year after. There is Lilly just got their drug, imlunestrant, approved about a month ago, in September 2025. And there's a bunch of other oral SERDs that are in clinical trials, including camizestrant, which was the basis for the SERENA-6 trial. I'm going to talk just a little bit about it in a second. But there are multiple other late-stage clinical trials that are happening. There's multiple guidelines that are recommending ESR1 testing in advanced breast cancer patients. In fact, NCCN updated their guideline in this year to specifically recommend ctDNA testing. And the reason for doing ctDNA testing will be clear, because what SERENA-6 showed was if you're actually monitoring, so currently the approved drugs are for, if you have progression, you test the patient for ESR1, it's positive, they're eligible for this, for these oral SERDs. What SERENA-6 did was they actually actively monitored these patients, testing them every 2 to 3 months to look for the emergence of ESR1. So this is where you're looking at molecular progression in advance of clinical progression. And for patients who had that molecular progression, if they were put on camizestrant, they had improved progression-free survival. And actually also, I don't have that graph here, but they also had improved quality of life as well. So really, this can be a practice changer. Again, there's been a few talks today talking about how like once this gets into guidelines and we get more approvals, this will really kind of change the practice of how you treat advanced breast cancer patients. So, but coming to a practical solution for that, so we have this kit called the DDPlex ESR1 kit. DDPlex is a set of assays that we launched with the ESR1. We're actually going to launch one with non-small cell lung cancer, which you'll hear about more tomorrow. But what we do here is we basically use a technology called blocker displacement amplification. I'm not going to go into the details, but the basis of it is that you're suppressing wild-type amplification, and therefore you can get higher sensitivity. So that's really the take-home message there. And so for this particular assay, we designed it with, initially we designed it with 7 mutations, which you can actually discriminate. So you can tell which particular variant within the ESR1 gene that you found. But then we heard from a lot of customers about 4 additional mutants that they were looking for, so we added this kind of supplementary assay to it. So now you can look at 11 variants, and together with those, they cover about 97% of ESR mutations in breast cancers. So we did actually, with collaboration with Biodesix, we did some analytical validation study, and this was presented at multiple forums. But if you do want to listen to this entire talk, so Lisa, who's right here, gave a really nice talk at the Digital Droplet PCR World about this analytical validation. And well, you can't click the link there, but if you're interested, we can send you the link. But what they showed here was that different inputs, you're able to get down to about 0.025% variant allele frequency with contrived samples here for all the 11 variants that you're testing. I'm going to talk very quickly about MSRE, which this is our approach for looking at methylation. So currently, the gold standard for methylation is bisulfite conversion, but that can result in degradation of the DNA, degradation of the input material. So using a restriction-sensitive, methylation-sensitive restriction enzyme overcomes some of those limitations of bisulfite conversion, and it also results in using small input of samples. So currently we have 50 designed assays that are on this digital portal that I talked about for detection of methylation. We are also building an engine whereby you can choose your favorite methylation site and it will design the assay for you. That'll be launched sometime next year. Really quick about some collaborations we have done with this. So this is with a group in Denmark where they design an assay where they're looking at methylation as well as single nucleotide variant together in lung cancer. So they were looking at the KRAS G12C as well as the methylation status of HOXA9. And they found a very good correlation between these two, especially also in various patient samples. Again, not going into the details of this talk, this is just to show that there's really good, that you're able to do both methylation and SNVs in a single assay, and also they show good concordance. We have had other collaborators who have actually combined the two and shown that if you combine the signals from methylation and SNVs, you actually get better clinical utility as well. But however, for this particular one, we do have a poster. It's poster G61. So if you're interested in more details, you can come to the poster there. I will end very quickly with MRD and monitoring. So basically what we are working on is a bespoke approach for looking at MRD, where we initially do a whole genome sequencing tumor normal of the tissue, and then we look at structural variants, but we can also do this with SNVs. We design those variants, and then they're manufactured by IDT or some other oligo manufacturer, resulting in an assay that can be anywhere up to about 16 assays per patient, and then you can follow the patient with that digital droplet PCR assay and look at whether they are ctDNA positive or not. We're doing, Gary talked a little bit about this, so this is part of the collaboration we're doing with Biodesix on MRD. We also have a couple of academic collaborations with Aarhus University and Allegheny Health Network here in the US to further develop this particular assay. Here's some results showing the LOD on this. So we did some, we designed an assay based on the database, and we looked at what's the LOD we can get with it, and we can get down to about 10 to the power minus 5, which is kind of the standard right now. And this also depends on where you do your cutoff on. Like if you're okay with higher false positive rate, your LOD can go down. So it's really kind of specific to the kind of application you're looking for. If you want high specificity, then you lose a little bit of sensitivity. So with that, that's the end of my talk. I did want to put a plug for, as I mentioned, we have another ddPlex assay that we're launching here at AMP, and there's going to be a talk about that tomorrow if you're interested. But again, thank you very much, and thank you for sticking here till the end. And yeah, with that, do I get to close, do an official closing? We close the meeting now.